Thank you so much for the opportunity to present on some of our work. My research focuses on the genetics of common obesity, and here I will present you how we use proteomics data to translate some of these gene discoveries into new biology. So first things first, the prevalence of obesity has increased tremendously over the past 40, 50 years. And of course, our genome has not changed, but the environment has changed. So people use these kind of graphs where we compare the prevalence of 1975 to 2016 to say that the genome is not important, it's the environment that is important. But that's only part of the study, because it's in particular individuals who are genetically predisposed who will gain the most in this obesogenic environment. So it's really genes and environment that contribute to the epidemic. And indeed, we know from heritability studies, such as twin studies and family studies, that heritability is reasonably high, varying between 40 and 70%, which means that of all the variation that we see between people in obesity susceptibility or in body mass index, 40 to 70% of that variation is due to the fact that we differ genetically, whereas 30 to 60% is due to the fact that we have different lifestyles. And indeed, when we do these genome-wide association studies aimed at discovering genetic variations associated with obesity, associated with body mass index, then we see a tremendous success over the past 15 years or so. In fact, in 2007, the first genome-wide association study discovered the first locus called FTO, and that's still the poster child of obesity genetics. And over time, the sample sizes of these genome-wide association studies have increased, and the number of loci has increased also. And by now, we have more than 1,700 loci that are associated with body mass index, obesity, fat percentage, and so on. And what we've learned from these discoveries is that genes that locate close to these genetic variants that are associated with body mass index, and we look at the expression of these genes and compare them with genes that lie outside these associated loci, then we see that the genes inside the loci are predominantly expressed in the brain, probably controlling the hedonic aspects of food intake, such as hunger, satiety, reward. So what I want to show you now, in the next 18 minutes or so left, how we use proteomics data to gain deeper insight in the biology. First, I show you a snippet on variant to function, then on what we call the Healthy Obesity Project, and then a study on life course genetics in the context of growth and weight gain over the lifetime. Okay, so this first snippet of research focuses on variant to function analysis, where we use proteomics to translate variant into new meaningful biology. And we start with a paper called, the latest paper from the GIANT Consortium, which is one of the largest genetics consortia that focus on the genetics of obesity. And they published in 2018 this paper that included 700,000 individuals, and they identified more than 500 genetic loci associated with body mass index. So our question was here, can we use omics data, including proteomics data, to translate each variant into the causal gene? So can we find the causal gene within each of these loci? And this project was led by my postdoc Virginia, who we performed the omics integration. So for each variant, she studied the association, of course, between the SNP and BMI, asking the question whether this association between SNP and BMI might be mediated through an association with gene expression, so between the SNP and gene expression, which then in turn would affect body mass index, protein levels, or metabolite levels. And what is key to these analyses, and I won't go through that into detail, but key is that the association between the SNP and body mass index and between the SNP and the protein levels, for example, co-localize, so that they come from the same genetic variant. So doing so, in her project, she combined data not only from Olink for more than 85,000 individuals, but also from SomaLogic and from MassSpec data. So she had pQTL data, meaning data where she links variants with protein levels for more than 140,000 individuals. She added also the expression data from across more than 47 tissues and 17 cell types. She then performed the co-localization analyses and found a large number of very interesting results. And this paper, she's still working on the analysis. So here I just give you a handful of examples. Take, for example, the first SNP out of these 5 is associated with body mass index. They are all associated with body mass index. But the SNP is also associated with protein levels and gene expression levels of these PPP1 R3b in oligodendrocytes, and at least that's for the gene expression levels. And digging a little deeper into the literature, this might point to a role of the glycogen metabolism. And you see a few other examples where we point to other metabolisms or other pathways, I should say, in other tissues, including sweet taste signaling, synapse integrity, brain xenobiotic removal, and another mechanism here with PPP1R3A is type 2 diabetes in lipodystrophy. She also found interesting results related to drug targets, and here's an example where this variant, RS17681708, is associated with higher body mass index and also higher type 2 diabetes. And the protein that correlates or associated with this same variant is GLP2R, which we know that companies are working on in the context of also gut health and maybe or predominantly gut health. And the target is here dapiglutide. The variant itself is again associated with intestinal dysfunction. It is also associated with lower glucagon levels, higher motilin levels, and lower secretin levels. So this together may tell you a little more about this variant being associated with body mass index and all the downstream pathways involved. So this is just one example on how we use proteomics to translate variant to function. A second story I want to share with you is about healthy obesity. Healthy obesity, and here the graph shows you a healthy fat cell and an unhealthy fat cell, but actually many more mechanisms are involved in healthy obesity. When we do gene discovery studies for body mass index or for obesity, we typically ignore the fact that not everyone who has obesity has type 2 diabetes. Not everyone who has obesity has hypertension and so on. So there are individuals who have obesity, but who are nevertheless otherwise have a normal or a normal cardiometabolic health. So in this study is again a gene discovery study, but not just for body mass index, but for body mass index in relation to other comorbidities or risk traits. So we studied body mass index, but also body fat percentage and waist-hip ratio in the context of these risk factors. And more specifically, we performed a genetic discovery study, a GWAS study, where we looked for variants that are associated with higher body mass index, but lower triglycerides. So this goes into what we normally expect because BMI and triglycerides are positively correlated. So here we were specifically looking for variants that uncouple BMI from triglycerides. So variants associated with higher BMI, lower triglycerides. And we not only did that for BMI and triglycerides, but we did all these combinations between adiposity traits on the left-hand side and cardiometabolic risk factors on the right-hand side. That way we did 24 by-trait analyses. We again performed a co-localization, like a lot of details that I don't have time to go into, but again pointing out that it's important. And we identified 205 loci with 266 association signals. So these are 266 uncoupling variants. And we made a score of these variants. So each individual carries these variants, but some individuals carries the adiposity increasing variant. So a high score would mean that a person has the predisposition to have high fat or high adiposity, but nevertheless low risk for cardiometabolic traits. So we generated 2 scores, actually. We have, based on these 266 variants, we have an uncoupling score, and then we have the typical body fat percentage score, or the fat score. We could say it's like the yellow one is the healthy obesity score, and the blue one is the unhealthy obesity score. And if we show the association of these scores with phenotypes in the UK Biobank. Then you see in yellow all the associations with the anthropometric traits, and in green as well. But you see that in yellow, the associations tend to be a little more attenuated. And just for orientation, here in red is the null line. So the main difference between blue, the unhealthy obesity score, and yellow, the healthy obesity score, is that the healthy obesity score had a more attenuated effect, but there is still effect on the adiposity score. And what I find particularly important is that the yellow, or the healthy obesity score, seem to have an association with a more favorable fat distribution. And the VAT-SAT ratio is an indication of that. The waist-hip ratio is also an indication of that. So that seems to be the main difference here between the healthy obesity score and unhealthy obesity score. And here you see their association with cardiometabolic outcomes. Again, in red is the null line, and again confirming that the score works. In yellow, the healthy obesity score is associated with protective effects, lower blood pressure, lower glucose, HbA1c, but higher HDL, whereas in green, you see the unhealthy score is associated with higher risk. This is just to confirm that these scores work. We did a series of analyses, but what I want to show you is the association of these scores with protein levels. Now, first, before I go into the association between the score and protein levels, I want to show you just the regular association between body mass index and the score. And we know that body mass index is very often, in fact, in 80% of the proteins, it's associated with body mass index. You see, it's like here, association between body mass index and all the protein levels. Standing out, of course, leptin, fatty acid binding proteins, but in fact, many of the proteins are associated with body mass index. So we asked, do the healthy obesity score and the unhealthy obesity score associate differently with protein levels? And here you see that there is an overlap. The unhealthy obesity score is associated with 915 proteins, the healthy obesity score with 337, and 208 of the proteins, there's an overlap. So if first I look at the 208 and then at 129, out of the 208 proteins, most of these are, 176 of these proteins are associated with both scores in the same direction. So I show you that here. So if we go to the right, the more you go to the right, the higher the adiposity. But for red is the unhealthy obesity score. So it means that the higher the score, the higher the adiposity, but also the higher the cardiometabolic disease risk. In yellow, the more you go to the right, the higher the adiposity risk, the lower the cardiometabolic disease risk. So what I show you here are all the protein levels that are associated in the same direction for both scores. So on the right-hand side, higher, on the left-hand side, lower. But you see leptin, for example, is high in the unhealthy score and high in the healthy score. So this actually means that the association with leptin is driven by fat, no surprise. So it's really not driven by the health, because then it would be leptin would be on the other side. It's driven by adiposity. Then 15% of the proteins are associated directly opposite way, and they are shown right here. So whatever shows up on the right in this graph will show in red, will show up in yellow on the left side. I'll give you an example. For example, in red on the top right, you see growth hormone receptor is higher in unhealthy obesity, whereas it's lower in healthy obesity. So here they suggest that these are all proteins that are not driven by the adiposity level, but by the health level of the score. And then there's another 129 proteins that do not show up in the unhealthy fat score, but only show up in the healthy fat score. And here it suggests that their association is driven by the health status. So knowing these associations, we get a better grip of where do these proteins play a role. We use these scores in the context of precision medicine because it's like now we have only two scores, we have healthy and unhealthy. But imagine that we have more refined, like when we come to healthy, more refined towards blood pressure or more refined towards glucose levels, then we can actually substratify the population according to their scores. And if we can substratify individuals based on their genetic score, then maybe we can also prescribe what kind of diet is good for them or what kind of lifestyle is good for them in this specific context. And what is good with genetics is that you can actually already do that at birth. Then one last snippet of my research focuses on growth and weight gain early on in life. And why is that important? Well, the genetic correlation between childhood and adulthood is very high, basically meaning that the genes that control body weight in childhood and adolescence are very similar, maybe even the same as the genes that control body weight in adulthood. However, when we look at the correlation between birth weight and childhood, then we see that actually the correlation is pretty low. So somewhere between birth and childhood, something happens that puts children on their BMI track. And once they're on that track, maybe from childhood onwards, they will probably stay on that track for a lifetime. So it will be important to identify what are the genetic regulators or the genetic predictors of this relationship between birth and childhood. So that's why in the projects to come, and we have no results on that, but we're generating the data, we will look at this life course effect, but we're focused on the first 20 years of life. One first study is that from the Danish National Birth Cohort. It's a birth cohort made of more than 60,000 mothers and their offspring. We focus on 15,000 or almost 16,000 mothers and offspring from the puberty cohort that have more detailed phenotyping between age 11 and 18. We have plasma samples of the mom during pregnancy at 2 time points. We have dried blood spots of the kids at birth from the PKU cards. And we will perform genotyping on all mothers and all offspring. And we will perform proteomics data on the blood samples during pregnancy of 2,000 moms and on the PKU cards of 2,000 of the offspring. Another birth cohort is that of the COPSAC, is also a Danish study. All kids, or 700 kids, but also their moms born in 2010. And we will perform proteomics at birth using the PKU cards, but also at 6 months, at 18 months, and at 6 years of age. And the kids are currently 13. They have very deep phenotyping, not only just your typical phenotypes, but also genetics, metabolomics. They have meal tests and so on. So we're very much looking forward to what these data will bring us. And I'll finish here, just highlighting the 3 topics I have described. And also, I'm very grateful to my team and my collaborations and also my funders. Thank you.